Hello everyone, my name is Zion Lamprou and welcome to my presentation about the role of punctuation in semantic mapping between BERT and Transformer model that was done as part of my research at the University of Strathclyde with the help of my two supervisors, Dr. Yasha Mosfegi and Dr. Frank Pollock. Natural language processing has been a task that is in continuous development in information retrieval. Modern NLP models seems to be learning something generic about the language. This property made it interesting to generate research around it and trying to interpret their inner representation and understand how they present language internally. This task is often hard to do because these models consist of millions of billions of parameters. Donovan Webber proposed a new novel approach that utilizes brain data to create a brain alignment between neural networks and human brain and tried to explain this way how these models represent language internally. In their experimental procedure, Donovan Webber tested 4 different models on how brain aligned they are: BERT, ELMo, UTE, and Transformer-XL. We believe that more BERT versions need to be tested on how brain aligned they are. Since treating punctuation in NLP is a very important topic, we wanted to test it whether treating punctuation in the text can affect the brain alignment between the human brain and the neural networks. Our experimental procedure can be divided into 3 phases. In phase 1, we extract features from a model we want to test on how brain aligned it is. Then phase 2 is to load back these extracted features and then train a ridge regression model alongside the brain data so the ridge regression model can predict those brain data. Then finally on phase 3, we're going to evaluate these predictions and determine how brain aligned this model is. Our experimental procedure consists of 3 different modules. The first module is the text dataset, the second module is the brain dataset, and then the third module is the model we want to test. The text dataset is chapter 9 from Harry Potter and the Philosopher Stone, and the brain dataset are pre-processed fMRI data provided by Donovan Webber, publicly available. Then we extended the set of models tested for brain alignment using Roberta, Distilibert, Electra, and Albert. We also tested 4 different punctuation scenarios on our experimental procedure. The first 2 scenarios involve changing the fixation symbol using the path token or the unknown token. That will make the models ignore this symbol when providing features for a sequence. Then, looking at the text, we found out that there are 3 different special characters: double dash, triple dot, and single dash. So we changed those with the path token as well, so the model will ignore them when providing features for a sequence. Then, adding to this set, we added the question mark and the dot, and then we put all 5 characters and then extracted new features from the model. Phase 1 of our experimental procedure can be divided into 4 steps. The first step is to extract the embeddings from the embedding layer if it exists in our model for every token in the dataset. Then, step 2 is to get the first sequence of S length and extract its features S time. This is done to give an alignment consistent between the fMRI data and the extracted features for every sequence. Then, step 3 is to extract the features from every sequence of S length in the text dataset. Finally, we save all the extracted features for every sequence for every layer in the model. Moving on, phase 2 of our experimental procedure can be divided into 6 steps. Step 1 will be to load the extracted features from phase 1 and reduce their dimension to 10 using PCA. This is done to keep a consistency between all the models, since every other model can produce a different dimension space. Then we time align the brain data with the extracted features in step 2. In step 3, we concatenate time points t-4, t-3, t-2, and t-1 together. This is done that we show by concatenating these time points together can give a better representation of what actually happens to the brain. Because the fMRI experiment was done in 4 runs, we need to remove the 20 and last 15 TRs of every run to combat the edge effect. After we pre-process our data, it's time to train the ridge regression model. We obtain a different lambda value for every voxel activation recorded for every subject, depending on which lambda value produces a lowest error score when training the ridge regression model. After we gather all our lambda values and the weights, it's time to make predictions. We use a 4 cross-validation settings since the fMRI experiment was done in a 4 run. Every run will be the test data of a particular fault. This experimental procedure for phase 2 is done for every layer in the model for every subject individually. Moving on from phase 2, we come to phase 3, where we evaluate our prediction. This phase can be divided into 5 steps. First step is to load the predictions for phase 1. Step 2 is to load the pre-computed voxel neighborhood provided by Toneva and Webe. The third step is to get a chunk of 20 TRs from our predicted data. Then we get the same 20 TRs from the fMRI data, and then another random chunk from the predicted data to serve as the wrong chunk. Then in step 4, we calculate the Euclidean distance between the predicted chunk and the correct chunk, and the predicted chunk and the wrong chunk. If the distance between predicted and correct is less than predicted and wrong, then this prediction is marked as correct. This procedure is done 1,000 times for every voxel recorded in the subject's brain. Then we obtained an average accuracy for every voxel that was recorded for every subject for every layer in the model. The first thing we did with our experimental procedure was try and reproduce the results from BERT reported by Toneva and Webe. On the left-hand side is the original BERT result reported from Toneva and Webe, and on the right-hand side is the reproduced results that we reported. As you can see, we were not able to reproduce the exact results as Toneva and Webe, but we believe our results are accurate since we use the same experimental procedure as the original authors. We also believe that they are accurate since the trend in the graph seems to be the same, with the peak to be a context length 10 and also the accuracy to decrease over time as the context length increases. We believe we didn't get the same results for 3 reasons. We didn't have the same hardware as the original authors. We also didn't know the exact procedure on averaging across every voxel, every subject, and every fold from the evaluation. And also, when performing PCA on phase 2, we didn't know the random seed that was used for the PCA. By using BERT as our baseline, we tested 4 different models and tried to determine if they are more brain-aligned than BERT or not. An immediate observation looking at the results is that all 5 models seems to be dropping accuracy as context length increases and also peaking at length 10. Looking closely, we can see that Roberta and DistilliBERT outperform BERT by a thin margin, and Albert and Electra are performing roughly the same as BERT. Using these results, we can hypothesize that the brain doesn't have a unified weight mechanism as Albert, but it has a shared weight mechanism as Roberta and DistilliBERT. But this is an assumption and it needs to be tested more. To test our 4 different punctuation scenarios, we used BERT to extract the features for every scenario and compare them against the BERT results when not reading punctuation in the text at all. As you can see, the accuracy of the ridge regression model doesn't drop as much when the context length increases compared to when we not read punctuation in the text. Also, the ending layer of BERT from 7 to 12 seems to outperform BERT when not reading punctuation. Moreover, layer 6 serves as a divisor between the starting and the ending layer of the model. We can hypothesize using these results that more context over to the brain, then more obsolete is the role of punctuation on understanding a sentence semantically. We can also hypothesize even though punctuation can help the reader skim through text faster, it might not contribute significantly to understanding the meaning of a sentence semantically. To summarize, we use the novel approach proposed by Toneva and Webe to create a mapping between human brain and neural networks to test how brain-aligned 4 different new models are. We show that Roberta and DistilliBERT outperform BERT by a thin margin, and also Electra and Albert perform roughly the same. We also tested 4 different scenarios to treat punctuation in the text before extracting the features from a model. We show that by treating punctuation, the ridge regression model accuracy doesn't drop as much when the context length increases when predicting brain data. We also show that the ending layers of BERT from 7 to 12 can outperform the BERT baseline with punctuation when not treated before extracting the features from the sequences. We also hypothesize that punctuation might not give significant information on the human brain on understanding a sentence semantically. As a future work, we believe that more models should be tested to determine how brain-aligned they are. We also hypothesize that the more context the human brain gets, the less significant the role of punctuation is to understanding a sentence semantically. We also hypothesize that punctuation might not contribute to semantically understanding a sentence. We also show an indication that the brain might have a shared weight mechanism similar to Roberta and not a unified mechanism like Albert does. These are just hypotheses and we believe they should be tested more to determine if they are true or not. Thanks for taking the time to see our presentation, and please let us know if you have any questions.